Namaskar. Today we will study Operon. This Operon is nothing but an arrangement where some functionally related genes are regulated as a unit with a common promoter, common operator and common regulatory gene. So what is this? Before that, let us first discuss that what is a gene. So gene is nothing but a part of your DNA. We know DNA is a double helical structure, and from DNA we get messenger RNA. So the information in DNA is transferred to messenger RNA through the process of transcription, and we need the enzyme RNA polymerase. Then the message present in messenger RNA is translated into proteins. The transfer RNA provides the amino acids and the required protein is synthesized. So the amino acids join and it forms the proteins, and required amino acid, the code is available with messenger RNA. So the gene is nothing but the coding part. Suppose say this is DNA and a part of DNA is our gene, and which is the coding part from which this messenger RNA can be synthesized. In prokaryotes like bacteria, what happens? There are some genes, say 3 genes, 4 genes, 5 genes. Together, these genes are functionally related. They together, we call them as structural genes, and there is an arrangement so that at a time all these genes together they produce the proteins. So if this operon operates, so here say this is the operon, and say three structural genes are there, A, B and C. So when RNA polymerase action will be there, from this gene protein A will be synthesized, from this gene protein B will be synthesized, and from this gene protein C will be synthesized. So the expression of these 3 genes A, B and C together will simultaneously will happen. So this is the function of an operon. So what are there in the operon? So there is one regulatory gene, a promoter, operator, the structural genes, then of course there will be a terminator, and some other sites are also there. Some activator sites and other sites are there, and I have shown here the catabolite activator protein site. So these operons are of 2 types. So before that, this gene again we can have 2 types, the catabolite gene or catabolic gene and anabolic gene. So basically the function of gene is to synthesize to express so that there will be synthesis of a protein. So gene expression will lead to synthesis of specific protein. So and that protein can be used for synthesis, biosynthesis. So these proteins basically nothing, they are the enzymes. So if these enzymes, say this is enzyme A, enzyme B and enzyme C, these enzymes are required for suppose synthesis of certain chemical, then we will call these genes as anabolic genes. And if these enzymes will be required for degradation of something, then we will call them as catabolic genes. So for those catabolite genes, there will be a catabolite activator protein site. Okay. And RNA polymerase is the enzyme which is required for conversion of DNA to messenger RNA. So RNA polymerase binds to the promoter. and the regulatory gene produces a repressor protein, and that repressor protein has affinity for the operator site. It binds to the operator site. So, when this RNA polymerase this will bind here, and this movement will be this words on this way. So, when RNA polymerase action will be there from gene A, gene B, protein B, gene C, protein C will be there. And RNA polymerase binds to the promoter and its direction, suppose it binds to the promoter and its direction will be this way. And if something will obstacle here at the operator side, then RNA polymerase cannot move, so there will be no protein synthesis. And who is that? This repressor protein. Repressor protein can bind to the operator site and block. Suppose something blocks here, so this movement cannot be there. And activator protein, this will bind here, so that it will facilitate the binding of RNA polymerase, and that will cause gene expression. So, two things can be there, either there will be gene expression or there will be no gene expression. So, gene will be on or gene will be off. So, two things can be there, on gene is on means transcription will be there and protein synthesis will be there. So, that is we call them a gene expression, and off, that means so no RNA polymerase action and no transcription, no protein synthesis, that is off. So, this in the similar way this operand can be on and operand can be off. So, though the condition in which the genes are actually normally on, that is a condition, and when the genes are normally off, that is one condition. So, we have some repressor proteins by the regulatory gene, and suppose say repressor protein now binds here. Binds here means there will be no gene expression. So, there will be no gene expression, that will be off. So, in that case, if some agent is there which can inhibit this repressor protein, then those genes will be expressed. And if some co-repressor will be there which will further consolidate the action of repressor protein, so then there will be no gene expression. Similarly, when the co-activator binds here and facilitates the binding of RNA polymerase with promoter, there will be gene on or the transcription of the genes. So, let us discuss first the two types of operon. So, inducible operon and repressible operon. We can have two types of operon, inducible operon and repressible operon. Inducible operon is normally off and repressible operon is normally on. And inducible operon is on when substrate is present, and repressible operon is off when the end product is abundant. So, here there will be biosynthesis. So, biosynthesis, so this will we call them as this repressible operon contains anabolic genes. So, there will be certain biosynthesis. For example, arginine biosynthesis, the operon is ARG operon. Tryptophan biosynthesis, the operon is TRP operon. So, when there will be sufficient arginine or sufficient tryptophan, that will turn off this operon. Similarly, here there is degradation, catabolism or degradation. So, lactose degradation, lac operon, arabinose degradation, ara operon. So, lactose we know is a beta galactoside degraded by the enzyme beta galactosidase. So, lactose is degraded to glucose and galactose. So, when lactose will be degraded to glucose, no lactose will be there, there will be off, and when there will be lactose, that will cause on. That means lactose, normally the operon is off, when there is lactose, so that is the substrate, when there is lactose, that will cause the operon on, and it will produce the enzymes like beta galactosidase which will break down the lactose. So that's why it is called catabolic. So let us see how they operate. So this is the example of your lac operon, and as we have discussed, this lac operon consists of three structural genes: lac Z, lac Y, and lac A. And lac Z produces the enzyme beta galactosidase, lac Y produces the enzyme beta galactoside permease, lac A produces the enzyme transacetylase. Beta galactoside permease and beta galactosidase, these two enzymes are required for your lactose metabolism, and beta galactoside permease is required for the permeation of lactose into the bacterial cell, and then by the action of beta galactosidase, lactose is degraded to glucose and galactose. So when there will be this operon will be on, there will be gene transcription, and from these three structural genes simultaneously these three enzymes will be produced, and this transacetylase this enzyme this is required. Actually it the bacteria gets rid of the toxic thiogalactoside because of this action. So here mainly for lactose we need these two enzymes, beta galactosidase and beta galactoside permease. Lac I is the regulatory gene which produces the repressor protein, and there is one catabolite activator protein site to which this catabolite activator protein binds. And catabolite activator protein is inactive itself, it cannot bind to the site, and to facilitate its binding there is a co-activator called cyclic AMP. Cyclic AMP first binds to the catabolic activator site, and then it can bind to the CAP site. So cyclic AMP binds to the catabolite activator protein, and then that will bind to the catabolite activator protein site. RNA polymerase binds to the promoter, and repressor, this repressor binds to the operator. So normally what happens, this lac I produces repressor, and repressor binds to the operator so that the promoter cannot move and there will be no transcription. So normally the operon is off. So lac operon is an example of inducible operon. Normally it is off. And why we call it as an inducible? Because when there will be lactose, that lactose will bind to this repressor protein. Lactose will inhibit this repressor protein so that this repressor protein cannot bind to the operator, and there will be a movement of this RNA polymerase. RNA polymerase can bind to the promoter and can cause the gene transcription, and all these proteins will be there. So lactose presence causes or induces the lac operon and causes the gene transcription. So this is an inducible operon. Lactose, there is on, no lactose, there is off. And so when the when lactose binds to the repressor protein and causes this gene transcription, and when there will be depletion of all the lactose, there is no more binding. So again the repressor protein becomes free and can bind to the operator. So the operon will be off in absence of lactose. And let us take another example of the repressible operon, that is the TRP operon. Repressible operon, why we call it as repressible operon? Tryptophan biosynthesis requires 5 genes: E, TRPE, TRPD, TRPC, TRPB, TRPA, and that means A, B, C, D, E. 5 structural genes are there, from which we will get the enzymes that will synthesize tryptophan. So normally it is on. So this operon is normally on because this is the regulatory gene which produces this repressor protein, but this repressor protein cannot bind to the operator. This repressor protein cannot bind to the operator. So here we have shown this is the site to which tryptophan binds. Tryptophan binding site, and there is another site which can bind to the operator, but this site is not open say now. So when this repressor protein is activated by tryptophan, that will change its conformation, and then only it can bind to the operator site and the RNA polymerase cannot move. So normally the operon is on and there is a synthesis of tryptophan because of involvement of all these 5 genes. But when there is synthesis of tryptophan will bind to this repressor protein, and repressor protein will block the operator site and no RNA polymerase action and no transcription. So that will cause off. So that's why it is called repressible operon. So no tryptophan, it is on, and when there will be synthesis of tryptophan, that will cause off. So these are the 2 types of operons, inducible operon and repressible operon. Thank you.